Alright, in this video we're going to talk about complete dominance and how to use a Punnett square to predict the phenotypes and genotypes based on two different parents, and this is basically Mendel's version of inheritance is what we're going to talk about today. So just to get us started, some terminology for the different genotypes, which ones are considered homozygous, heterozygous, and if we've got a big H, little h, this one would be considered heterozygous. And remember, hetero means different, so we've got a big dominant trait and a recessive trait. If there's two that are the same, it is called homozygous. And because these are both dominant, it's going to be called homozygous dominant. And this one would be called homozygous as well, because they're both the same. But this would be homozygous recessive. Excuse my handwriting. Okay, so what we're going to do is we're going to use what we know about Mendel's idea of complete dominance to answer certain and predict certain chances of having offspring that will have freckles or will not in this example. So before we get started, the steps to always do one of these problems correctly, always do these 4 steps. And if you do them and you write them down, I almost guarantee you're going to get the right answer every time. So really the first thing is make a key, or really write down everything, all the information that they give you. And so in this example it says in humans freckles is a dominant trait. So what I'm going to do is I'm going to make a big F for freckles. That's supposed to say freckles. And a little f for no freckles. So I did step 1, I made a key. The next step is to figure out, well, what the parents are, to figure out what the children are going to be. And so if a freckled male and a female are both heterozygous and they both had a baby, what are the chances their offspring would have freckles? And so it says the male and female are both heterozygous, which means they both have a big F and a little f. And in complete dominance, in this version, it doesn't matter if you put the male or female on the left or right. It doesn't matter at all. But what I want us to stop and ask ourselves is this. How many genes does everyone have for a trait? 2. How many parents do you have? It's 2. And each parent gave you one version for each trait. And so you have 2 versions of every trait. And this individual right here also has 2, as they should. And my question to you is, well, how many are they going to pass? Well, they always pass 1. And do we know which one they're going to pass? The big F or the little f? And the answer is we don't know. And so that's why we make a Punnett square. We don't know which version or which allele they're going to pass for a certain trait. So we make a Punnett square. And what we do is, because we don't know which one they're going to put, we're going to put each one in a different box. Just like this. And you don't always have to write this out, but I'm going to say this is dad up here, and this is mom right here. Which means that really what we're saying in a Punnett square is what these individual boxes represent is the one gene that dad will pass for every trait that he has. And the reason, or the process in which you have only one trait that gets passed, is meiosis. So really what's in these boxes is not just letters that randomly mean, freckles. Really what it is the gene in each one of the sperm for the dad. So these really are where the sperm, the sex cells. And what's in here is going to be the eggs. And so what we're showing when we do a Punnett square in the middle is going to be the first cell of the next generation, or the cell that gets that's created when a sperm and egg fuse together, and that's a zygote. So in this first place right here, we have big F, we have big F, and this is what the zygote will look like. This is the genes that this individual will have in its zygote, big F, big F. Here we have big F, little f, big F, little f. And again, I'm just taking this F here, this little f here, and these are going to be all the possibilities that they join. This F will come down here, this F will come over here. And a lot of students ask me, does it matter if the capital F goes first? or lowercase f, and I would say always put the capital letter first. So always put the capital letter first. So my point here with this picture is the Punnett square is not just something we do. It's not an answer to a question. The Punnett square is a tool that we use to figure out what the probabilities of the different zygotes are going to be. And so when we make our Punnett square, we still haven't have our answer. We're going to write down our answer is really going to be our genotype and our phenotype ratios. So the first genotypic ratio is going to be basically we've got one big F, we've got two big F, and then we've got one little f, So our ratio is going to be a 1 to 2 to 1, big F to big F, to big F to little f, to little f to little f. Okay, so that's our genotypic ratio, just what the genes are. And then we want to ask ourselves, well, what is the phenotypic ratio? And in complete dominance, there's only two different phenotypes. You either have freckles or you don't. And so I would say that if you have a big F and it's always going to be completely dominant over the recessive trait, there's going to be 3. So we've got freckles versus no freckles. And it's going to be a 3 to 1 phenotypic ratio of freckles to no freckles. And so now that we've done our Punnett square and we put together our phenotypic and genotypic ratios, whoops, it asks what are the chances our offspring would not have freckles? So what are the chances our offspring would not have freckles? And the answer is, it's a 3 to 1 ratio. So out of 4 possibilities, one of them will not have freckles. So that would be like 1/4 or 25%. So then we have our answer and we circle it. Okay, so that's how we answer one of these problems. And just to reflect a little bit more, going crazy. What is a Punnett square and what does the Punnett square represent? Just as a quick recap. The first thing you need to remember is this. Parents have 2 genes for every trait, and they're only going to pass one, and it's random. You can't control which one you're going to pass. So what a Punnett square is, a Punnett square is a visual representation of all the possible genotypes that their offspring could have. That's what a Punnett square is. I hope this video helps. Thanks for watching.